I'm Eric Roberson from the Center for Neurodegeneration and Experimental Therapeutics and the Alzheimer's Disease Center in the Department of Neurology at the University of Alabama at Birmingham. This paper in Brain is a collaboration between my lab and Harry Sontheimer's lab in the Center for Gliobiology and Medicine and the Department of Neurobiology at UAB. In this paper, we investigated the effects of vascular amyloid on the structure and function of the gliovascular unit. Amyloid beta peptides are the main driver of Alzheimer's disease. And in affected patients, they accumulate in two different locations, in the extracellular amyloid plaques between brain cells, and along the cerebral vasculature in a form called vascular amyloid. We know that vascular amyloid predisposes vessels to leakage and to intracerebral hemorrhages. Our question here was how vascular amyloid might affect the regulation of cerebral blood flow by the gliovascular unit, comprised of astrocyte end feet that surround blood vessels and transduce nearby neuronal activity into changes in vessel diameter by release of vasoactive regulators onto pericytes or vascular smooth muscle cells. Prior work from Dr. Sontheimer's lab had shown that brain tumor glioma cells could separate astrocyte end feet from the vessel wall and limit their ability to regulate vessel diameter. We wondered if vascular amyloid might have similar effects. In these studies, we used the J20 line of amyloid precursor protein, or APP, transgenic mice, using a variety of imaging approaches to assess the structural and functional impact of vascular amyloid.
I'm Ian Kimbrough, one of the lead authors of this paper. The first thing we found with the vascular amyloid deposits in APP mice was the physical displacement of astrocytic end feet by vascular amyloid. In these images, the vessel wall is labeled with a blue Alexa dye, the vascular amyloid green, and the astrocytic end foot has been loaded with the red ROD2. You can see how the vascular amyloid intercalates between the end foot and the vessel wall, just as we'd previously seen with glioma cells. We also observed the same thing in electron micrographs of vessels from APP mice. Next, we investigated how this would affect the ability of astrocytic end feet to regulate vessel diameter functionally. We used pharmacological stimuli to activate either astrocytic end feet or vascular smooth muscle cells in acute brain slices. Imaging vessels with time-lapse microscopy revealed a diminished vessel response in slices from APP mice. To be able to actually visualize vessels that we could confirm were affected by amyloid, we specifically labeled vascular amyloid in living mice using the dye methoxy-X04. This allowed us to visualize the vascular amyloid and determine how it might impact the function of the associated blood vessels. High-resolution 3D images demonstrated that vascular amyloid developed in ring-like structures around the vessel circumference, essentially forming a rigid cast or exoskeleton. We directly tested vessel response to astrocyte stimulation using laser pulses to uncage calcium in astrocyte end feet surrounding vessels in ex vivo brain slices. As you can see, calcium uncaging in astrocytes leads to a rapid constriction of vessels in this non-transgenic animal. However, stimulating the astrocytic end feet around a vessel affected by vascular amyloid in APP mice produced a much smaller change in diameter. We then wanted to see if the same effect would occur in vivo in pressurized blood vessels and in the presence of blood flow. For these experiments, we performed multi-photon in vivo imaging through a cranial window and adapted a novel, label-free optical activation technique that allowed us to activate vascular smooth muscle cells with a pulsed laser through an intact meninges, which, as you can see here, induces vasoconstriction in unaffected vessels. However, in vessels with vascular amyloid, we saw a dramatic reduction in vessel response. Vessels were not able to constrict where the amyloid was present. Interestingly, when we imaged vessels where the amyloid was interspersed along the vessel, we found that segments with vascular amyloid had limited constriction, while vessel segments that were not affected by vascular amyloid responded normally, suggesting that, apart from the physical restriction created by the vascular amyloid, the ability of vascular smooth muscle cells to constrict the vessel was intact.
This study provides evidence that vascular amyloid impairs both the structure and function of the gliovascular unit. The presence of vascular amyloid physically displaces astrocyte end feet away from their normal location around the blood vessel wall, and at the same time it creates a rigid cast around the vessel that increases its rigidity. The functional effect of these structural changes is a limitation of the vascular reactivity of the vessel and a limited response to the gliovascular unit. I think that these changes emphasize and contribute to our growing appreciation of the vascular contributions to Alzheimer's disease and posed several questions for further research, including how these changes might contribute to the loss of cerebral blood flow that occurs in Alzheimer's disease, and how these abnormalities might be affected by the kinds of anti-amyloid therapeutics that are now in clinical trials.